Andrea, thank you very much. You pretty much announced the next speaker. So about the same thing, but in mouse models from our Duško Lainšček. He comes from the, I would like to say, really phenomenal department for synthetic biology and immunology, and which I'm a part of with this CTGCT, which is a new project. Duško, happy to have you on the floor.
Thank you, Gregor, for your kind introduction. And by the way, you're doing an excellent job.
Thank you.
First of all, I would like to give a warm welcome to everyone, especially to parents who are basically fighting this battle. And I would also like to congratulate Spela for putting this beautiful event together and also for being the driving force for this astounding thing. As already Andrea beautifully presented what they did, what a wonderful journey also they embarked on. I would like to present you basically our team effort in order to evaluate the effect of the leading candidate for treating basically CTNNB syndrome on a mouse model. You all know that in new drug development process, there are still crucial parts where you have to do some preliminary preclinical or non-clinical testing on in vivo in order to basically establish all the pipelines and also to get approval for human trials. And we usually use mouse model because of the great similarity of the genomes between the human and mouse species. And also they're relatively easy to work with, and basically also to some sort of degree, you can easily translate the findings from the mouse model to humans. In this story, we basically developed a new CTNNB mouse model. We then conducted a history study of animals where we basically characterized the animal. And then by connecting different research facilities, we also conducted efficacy study, and then also subsequently in vitro validated the therapeutic transgene effect of the candidate that was before presented by Andrea. We conducted a non-GLP, non-clinical or preclinical efficacy study, but we also have to take into account that animals will be also used for later on GLP toxicology study, which will then be beautifully presented by Jan afterwards. So getting and having a good animal model, in our case, mouse model, is quite important. So the colleagues at Jackson's laboratory basically developed a so-called heterozygous CTNNB1 gene model, mouse model, where basically they mated beta-catenin flocs with SOX2-CRE mouse. That basically led to loss of function model, meaning that only one allele of the CTNNB1 gene is presented in these animals. And afterwards, when the genotype of the animals was confirmed, we then carried out so-called history study, or we characterized the animals with some testing. Firstly, we discovered that there's a significant difference in body weight between the wild-type littermates and CTNNB mouse model. You can see here differences for the males and females. So we are quite happy that we already observed some phenotypic changes in our mouse model. And then another simple test was carried out using so-called rotarot. Basically is a assay that basically determines the motor coordination or balance of the animals. You basically put mice on this rotating spindle and you basically measure the time that allows the animal to basically stay on that. And we also discovered that at 12 weeks of age, we observed that for the males, that they drop more frequently or faster compared to the wild-type littermates, again, confirming that there is a phenotype present in our disease mouse model. After obtaining this mouse model, we then went further and we contacted Charles River, basically a connecting point of the Jackson's laboratory, where they conducted a non-GLP efficacy study in order to basically pinpoint the therapeutic effect of the leading candidate that Australian people built. So they injected, they injected AAV9 CTNNB1 vector into the mouse brain. Here, ICV injection, they injected 5 microliters of the virus, basically by doing stereotactic injections. And we use, as you can clearly see here, 5 different groups of animals, whereas wild-type animals were used as a control. And then the second group was basically heterozygous animal receiving placebo treatment. And then we use low dose, mid dose, and high dose of treatment for the animals with the appropriate CTNNB syndrome drug that is being now developed. The animals were injected at the age of 4 weeks, and then at the age of 12 weeks and 20 weeks, several different behavioristic assays were conducted, for instance, open field marble assay, kinematic gain analysis, and so on, to really pinpoint the therapeutic effect of the drug. I will just present you the main findings here because, as you saw before, many different tests were carried out. And I would just like to highlight the test that showed statistical significance between the treated animals and non-treated animals. First test that was employed was so-called marble burying test. Basically, this is a very nice, cute assay where you basically have a cage filled with bedding material that should be familiar to the animals, that they don't afraid of this new bedding material. And then you just put marbles on that bedding material. And basically, this marble burying test determines the level of depression or anxiety when the animal encounters a foreign object, because these marbles are foreign objects to them. And you also basically can model compulsive behavior to those animals. And then after defined period of time, I'm thinking we're talking here about 30 minutes, 1 hour, you basically just count the marbles that the animals were buried. And you can clearly see here that we observed statistical significance between the non-treated animals and treated animals, again confirming that we already observed a positive therapeutic effect on our mouse model after given the new drug developed for the CTNNB1 syndrome. The second assay that was employed, the so-called open field test. Basically, you put animal in an open cage, and then the camera basically films the animal's movement. And you can clearly see that if you have a so-called normal mice are very intrigued, and they basically investigate their surrounding. Whereas if you have an anxious mice, they don't like to investigate foreign surroundings, and they like to stick to the edges of these new cages. And basically this open field test is tested to use it to determine locomotor or anxiety-like behavior. And again, we were quite happy when we discovered that, there was again a statistical significant difference between the treated animals and non-treated animals, confirming that this drug is actually doing something positive. The last assay that also showed significant improvement, and we were quite happy about that, was so-called fine motor kinematic gait analysis, which is basically an automated high precision system to quantitatively measure changes in gait and balance, which are basically the clinical endpoints in movement disorders. And here you basically put animal in this corridor, like Laurent was before presenting for their device, and then the camera measures fine movement of the animals, movement of the paw trajectories, movement of the tail, hind limbs, and everything on. And then the computer basically translates those measures into definite scores. And here we were basically observing general gait pattern parameters, body posture and balance, and also fine motor skills. And we were quite happy when we discovered that there was again statistical significance at the age of 12 weeks, 20 weeks in all females and males for the treated animals, meaning that, again, positive effect in the phenotype, in the behavioral, in the motor function of the treated animals is present also here. And here we discovered that for the tail and back body posture, swing, and also paw trajectories, these are the crucial parameters that we observed that were improved. And we can confirm here that by doing 3 different statistically significantly important assays, we can conclude that this new drug has potential to be considered as a cure. And then afterward, at the end of the experiments, of course, the animals were humanely sacrificed, euthanized, and all the tissue were collected. And then qPCR was used to basically determine the level of the CTNNB gene expression from those viral particles that were injected in the mouse brain. And you can clearly see here a dose dependence in all brain tested regions, confirming that actually the vector is doing its job, meaning that the CTNNB gene, transgene, human transgene is being expressed in all treated animals. Okay, we observed that we could influence higher CTNNB1 gene production, which is basically the main thing of injecting the virus into the body of the patient or here animal organism. But we were keen on finding if that higher expression of the CTNNB1 gene can also result and translate to the higher beta-catenin production. If we observe higher protein production, because the proteins are those ones that are responsible for normal cell proliferation, normal cell function, which are basically the thing that drives you and also that are responsible for your life. And we were quite happy when we discovered that basically in all cases, we tested subcortex, cerebellum, hypothalamus, all different parts of brains. And we discovered that we observed higher beta-catenin production compared to untreated controls. And we were quite happy with those results. And we also confirmed, I don't know if you see this also by immunohistochemistry. Here's the picture of the mouse brain. And you can here see with yellow, the expression of beta-catenin protein. At the end, we also wanted to some sort do a so-called safety profile measurements. So we were wondering what happens with peripheral tissue. Therefore, we analyzed muscle tissue and liver tissue, as Andrea beautifully pointed out, that these are the off-target effects that you don't want to basically hit with your AAV9 treatment. And you can clearly see here that there was no effect of increasing beta-catenin production in this off-target tissue, meaning that this cure is considered here safe. And also we conducted a blood biochemistry panel. We observed, basically, we checked for kidney functions, for liver enzymes, for metabolites, and we didn't, again, observe any detrimental effect of the treatment of those animals, again, confirming safety or no side effects of the treated animals. So to summarize my talk, in this collaborative team effort, we basically developed a new mouse model to study CTNNB1 syndrome. We showed positive effect of the treatment. We observed not only higher DNA expression, we also observed what is also the most important thing, higher protein expression, no adverse effects were seen, and we can conclude to some degree that this is a promising clinical strategy for all the CTNNB syndrome patients. So at the end, I would like to acknowledge and thank all the people that are listed here. And of course, I would like to thank you for your attention.